So we're going to be talking about two experiments today. One is the gradient plate for the isolation of streptomycin-resistant mutants, and the other is the bacteriophage plaque assay. But we'll start with the gradient plate first. And so, as you've already learned, random chance mutations occur in bacteria, and those mutations enable them to resist antibiotics. And as it turns out, in any given population of bacteria, there's a probability of finding a few cells that have those mutations that make them resistant to basically any particular antibiotic. And so even before human beings started using penicillin back in the 1940s, there were already bacteria that had some resistance to it, even before humans started using it. So that's an important concept, that there's always some resistance present even before you use an antibiotic. And so what we're going to do today is show that experimentally by something that you would have done in class. And remember, we also saw this kind of thing in the Harvard video, which had that large 4-foot rectangular petri dish. And we saw E. coli mutate and change and become more resistant over a period of about 11 days. And so we do a much simpler experiment to demonstrate that there's already some resistant bacteria present. And we're going to be using E. coli and the antibiotic streptomycin. Who remembers, what does streptomycin do? What's its target?
Is it narrow spectrum?
It is, but what's the target inside the cell? What's the specific target? Everybody's forgotten all about their antibiotics. Any guesses? Like the ribosome, right? Translation, protein synthesis. So we're going to be using streptomycin and E. coli, and we're actually going to create a concentration gradient of streptomycin. So we'll have one end of the agar plate is a very high concentration, and it decreases as you go across the agar plate to the other side, where there's little or no streptomycin. And the way we do that is by using 2 layers of agar. So each group would have gotten 1 sterile petri dish. Then you take one of the melted agar deeps in the water bath, and you pour that into the sterile petri dish, put the lid on, and then put a pencil under one edge of the dish. And so the idea is that the agar will cool and solidify at an angle. So when you take the pencil out from under, you have agar at an angle like we see here. Then you take the second melted agar deep, you add your streptomycin solution to that. And we put green food coloring in the streptomycin solution so we know where the streptomycin is. And so you pour your streptomycin agar on top of the first layer. So you create this double layer plate, 1 green layer and 1 plain layer. And so the idea is, on this end here, you see where it's all just the green streptomycin agar, that's where the highest concentration is. As you move across the plate, the streptomycin is going to diffuse into the plain agar layer, which means less and less streptomycin is present on top. And so the concentration of streptomycin decreases as you move to the other side of the plate to where there's little or no streptomycin at all. And then we spread some E. coli on the surface, incubate the plate, and observe where the E. coli have grown. What we see is something like this. And this is an actual plate that we got one semester. And so where it's dark green, that's the highest concentration of streptomycin. And so that would be on this end. As we move across the plate, there's less and less streptomycin until we get to a point which is basically none. When you look at the E. coli, you see this very thick confluent growth here where there's no streptomycin. As you move across the plate, there's less and less E. coli, right? The growth gets thinner and more spread out, and eventually you're left with individual colonies. Remember, each colony came from a single cell. So this handful of colonies here represents just a tiny fraction of 1% of all the bacteria you put on that plate. Most of the E. coli had been killed. A few survived to form these colonies. So those that have survived are resistant, right, to the concentration that's on that plate. So what we've demonstrated is that in that population of E. coli, in that culture that we used, there were already some cells that had some resistance to streptomycin. Even though it's just an ordinary culture of E. coli, we don't do anything special to it. We just grow it as we need it, and then we spread it on the plate. And what do we see? We see some resistance there. Not a lot, right? Just a tiny fraction of 1% of all the cells on that plate, but there are some resistant bacteria present. Does everybody understand this picture here and what it's showing us?
Please, can you explain it again, please?
So remember, we have two layers. One layer contains the streptomycin and is green, and the other layer is just the plain color of the agar, which is kind of yellowish. And so where it's darkest green, you have the highest concentration of streptomycin. As you move across the plate, there's less and less streptomycin until there's none. And then we spread the E. coli on the plate, let the E. coli grow. And what we see is very thick growth on the part of the agar where there's no streptomycin. As you move across the plate, there's actually less and less E. coli. Right? The growth starts to thin out. And then you end up with some individual colonies growing in the high streptomycin concentration, and these colonies are resistant E. coli. It's just a very small number relative to all the cells we put on the plate, but there are some naturally occurring resistant bacteria there. And so we just demonstrated that in that population of E. coli, there were some naturally occurring resistant bacteria. Most of them, no, but some of them, yes. And so then why would you expect to find any streptomycin-resistant mutants? I keep saying that there are some there, but why?
Maybe because they just decided to mutate?
Well, they don't decide to mutate, right? The answer is in red. What's the answer to that question? Why would you expect to find any streptomycin-resistant mutants?
Oh, it's a spontaneous mutation.
Yep, random chance spontaneous mutation, just a random thing. And that mutation does not serve any purpose until the bacteria are exposed to streptomycin. And then those resistant mutants have an advantage, right? They will survive, they will grow, they will reproduce. But it's all random chance spontaneous mutations. So in this experiment, what is the purpose of the streptomycin in this experiment?
To select for resistant mutants. So to
Right.
select E. coli that can survive.
Yep. So the streptomycin is our selection agent. We're using it to select for those naturally occurring resistant mutants. And the reason why we know they're there is because they're growing in the high streptomycin concentration. So streptomycin, we're using it to select for resistant mutants. Does streptomycin cause mutations to occur?
No.
No, it's merely selecting for those mutations that are already present, they're already there. And then what are some of the possible mechanisms a mutant might use to circumvent or to resist the streptomycin? So we talked about several possible mechanisms in lecture. Does anyone remember some of those possible mechanisms? So how do bacteria resist antibiotics?
Oh, they can destroy the drugs with enzymes.
Right, they might produce an enzyme to destroy the antibiotic. What else?
They can also change their target site where the drug would bind to.
Right, there could be a mutation in the target sites. Anything else?
You can eject the drug, rapid ejection of the drug.
Right, so remember the efflux pumps, which pump stuff out. So there are many different possible mechanisms, right, that allow bacteria to resist the antibiotics. So any questions on gradient plates? Nope, all good with gradient plates? Okay, so for the bacteriophage plaque assay, there are actually two handouts. We'll look at my version, which simplifies things, and then we'll look at the other version, which has more detail, more background information, and some questions, and even one problem. So we already know that bacterial viruses infect bacteria. In this case, we're using a T-even bacteriophage, so one of the spaceship, right, viruses that lands on the bacteria, right, to infect them. And we're using either T2 or T4. And then our host is E. coli B. And we're using E. coli B because it is particularly susceptible to infection by T-even bacteriophages. So that's why we're using E. coli B. Now, in order to evenly mix the phage and E. coli together, we're going to use something called soft agar. So soft agar is just a much thinner agar. It melts more easily. We can keep it liquid more easily. And so we use the soft agar just as a vehicle to mix phage and E. coli together. And then we'll pour that mixture on top of an agar plate, gently swirl the plate to spread out the soft agar. It will quickly solidify, and then we can incubate the plates. But we must use soft agar as a way to mix the phage and E. coli together, right, to get a nice even mixture. So all this stuff will be kept in a 47 degrees Celsius water bath. So when we incubate our plates, the bacteria, the E. coli will grow, they'll cover the entire surface of the plate, and the viruses will reproduce and kill the E. coli, leaving holes in the growth. And of course, we call those holes plaques. Once again, we're going to count plaques. Our number will be between 30 and 300. We will calculate our serial dilutions, our dilution factors, and determine PFUs per milliliter. And a PFU is a plaque-forming unit. And a single virus particle, also known as a virion, right, creates a plaque. So it's exactly the same as what we did when we diluted our bacteria and counted colonies. The exact same procedure, except that we need to use a host, E. coli, to grow our phage. But otherwise, all the dilutions, all the calculations, everything is the same. So here we see an example of one of our plates. You notice that there are holes in the growth, right? The E. coli is kind of a tan color, and then the grayish holes are the plaques. So we're actually going to count them. Now, the plaques are full of virus, right? Because all the cells have been killed. And why did we say that the plaques are going to stay small? Why don't the viruses keep multiplying and destroy all the bacteria on the plate? What was their reason for that?
To infect the log phase.
Right, because the viruses want log phase bacteria, right? Very high metabolic rates, high rates of reproduction. And under ideal conditions, bacteria can start entering into stationary phase in about 8 hours, soon to be followed by death phase. So the viruses stop infecting because it's no longer advantageous to the virus, right, to infect those kinds of cells. So the viruses just sit there and wait until better hosts come along. What does TNTC mean?
Too numerous to count.
And TFTC?
Too few to count.
Right, too few to count. Sometimes instead of saying TNTC, they may write TMTC, as in too many to count. So sometimes you may see TMTC, but originally it was TNTC, too numerous to count. So because we see plaques, what kind of life cycle is that for the virus? Because we see plaques, what life cycle is that?
Probably lytic because it's killing them.
Right, yeah, so it's lytic. What would we see if we were using a lysogenic phage? So if we had used a lysogenic phage, what would we be looking at right now? A lysogenic phage. what would we be looking at?
Would there still be a full plate of agar?
Oh yeah, the
nothing?
agar is there, but what would you see on the plate?
No holes?
Right, so just E. coli, right? Just a plate covered with E. coli. In fact, you would not know that there was any phage at all, right? Because the lysogenic phage has a dormant prophage, so there's no way to see that. And so all you would see is just E. coli covering the surface of the plate. And so that's why we use a lytic phage, so we have something we can count and determine the concentration of. So normally, all the soft agar deeps are in a 47 degrees Celsius water bath, and then you also put all of your broth tubes to do your dilutions in the water bath as well. And the reason why, after you finish doing your dilutions, you're going to transfer 1 milliliter of each dilution to a soft agar deep. If your broth is cold and you add it to the soft agar, it's going to make the soft agar turn thick, and then you can't pour it or spread it. So we want everything to be warm, right? So that way the soft agar does not thicken. And even the TSA plates we're going to pour on top of, we keep those in the incubator so they're nice and warm, because if you pour soft agar onto a cold agar plate, it immediately solidifies and you can't spread it. So we do keep everything warm in this experiment so we can spread our phage and E. coli mixture more easily. So every group starts off with a phage that's already been diluted down to 10 to the minus 4. So you're already starting off with a dilution. And then you get 4 more 9 milliliter broth tubes. So you can do your dilutions into the 9 milliliter broth tubes. So in this experiment, it's actually much simpler to do all the calculations because everything is 1 milliliter. All the transfers are 1 milliliter. The amount you're going to ultimately put on the agar plate is 1 milliliter, so that makes all the calculations much simpler here. But we're adding 1 ml into 9, so that's a 1 to 10 dilution. What's 1 to 10 exponentially? What's 1 to 10 exponentially? One divided by 10 equals what?
0.1.
0.1 or 1/10. And so what is that in exponent form?
1 times 10 to the negative 1.
All right, so 1 times 10 to the negative 1. And remember, since we know that everything is 1 times, 10 to whatever exponent, we don't have to say 1 times. We can just say the exponent. So it's 10 to the negative 1. So we're doing a 10 to the negative 1 dilution of a solution that's already diluted 10 to the negative 4. Right, so negative 1 and negative 4 make
Negative 5.
negative 5. And then we mix up that tube nice and thoroughly. So we transfer 1 milliliter from that tube to the next tube. And we're doing another 10 to the negative 1 dilution. But we're diluting a 10 to the negative 5 solution by 10 to the negative 1. What's our total dilution?
Negative 6.
Negative 6. And we repeat negative 7. And again, negative 8. So those are all the dilutions in the tubes. Then we're going to transfer 1 milliliter of each dilution to the corresponding soft agar deep. Remember, this is all ultimately going to be poured on top of an agar plate, so we're just adding 1 milliliter. And because it's 1 ml, the dilutions are not going to change, right? Because it's just 1 ml. 1 times anything or anything divided by 1 is the same thing, right? So you add your 1 milliliter of each dilution to the corresponding soft agar deep, and then you add 0.2 milliliters of E. coli B to all the soft agar deeps. Because remember, the purpose of the soft agar is just to mix the phage and E. coli together. And so 0.2 milliliters, you guys probably haven't been able to get much experience pipetting. It's analogous, 0.1 milliliter is actually approximately 1 drop. And so if you added 2 drops, that would be 0.2 milliliters. Or on the pipette, you would actually measure from the tip up to 0.8. Because remember, it counts backwards from the tip up to 0. And just add that to each tube. Mix that up, and then you pour that on top of the TSA plate. Swirl gently for about 10 seconds to spread out that mixture across the surface. In a few minutes, the soft agar has solidified, and then you can incubate those plates. So in this case, the effective dilution on the plate is the exact same as what it is in the tube, because we're transferring 1 milliliter. If we were doing 0.1 milliliter, that changes our dilution on the plate, right? But we're just doing 1 ml, so right, the numbers on the tubes, right, 10 to the negative 4, 5, 6, 7, 8, are the same on the plates this time. Now we know the number that we need is the dilution factor. Remember, technically that's 1 divided by the dilution. 1 divided by a negative exponent becomes a positive exponent, right? So 10 to the 4, 5, 6, 7, 8 are all the dilution factors. So you examine the plates. Your number of plaques needs to be between 30 and 300. Count those plaques, multiply by the dilution factor of the plate, and then put the number in proper scientific notation. So I've given you an example. There were 147 plaques on plate number 4. Right, plate number 4 has a dilution factor of 10 to the 7. So 147 times 10 to the 7 in proper notation is 1.47 times 10 to the 9. So remember, we made the 147 smaller by 2 decimals, so we got to make the exponent bigger by 2. And it's PFUs per milliliter, or plaque forming units, which are virus particles. It's the number of viruses per milliliter. And does anybody know the sort of fancy word they use for concentration when it comes to viruses? They have a separate word they use instead of concentration, and it's a smaller word. Anybody have any idea what that word is?
No, but probably once you say it, I would have heard of it.
Have you ever heard of titer? T-I-T-E-R, titer?
Yeah, I've heard of that.
Right, so titer just means concentration. So it's another word they use sometimes, and usually when they're talking about viruses, they use titer. But T-I-T-E-R is the same as concentration. So that's our concentration. Now what's interesting about this lab is that since each group had the exact same 10 to the minus 4 concentration to start with, when you finished and you did your calculation, you should all have been pretty much the same. And that's where we can see how good or bad a group's technique is, right? If all the groups are pretty much the same number except for one group, we know that one group had bad technique, right? Lucky for you guys, you're not doing this, so we can't see who has the good technique and who doesn't. But that's the main idea. And so let's look at the other handout, which has a lot more background information, a lot of which we already know because we know about bacteriophages. You know the 5 stages of replication. And so again, we have our phage plates, lots of E. coli or whatever the bacteria are growing on here, and then the holes in the growth, which are the plaques. And of course, if you see plaques, that means the virus is following the lytic life cycle. Now here we see a slight difference in the equation. It's because I like to include the volume that we put on the plate included with the dilution calculation, so the dilution factor is all set and ready to go. But the other way to do it is at the end and divide by the volume you put on the plate. So in this case, we're only using 1 milliliter. So if you divide by 1, nothing changes, right? So in this experiment, it's basically just the number of plaques times the dilution factor on the plate. But like I said, I normally include the volume on the plate as an exponent and then factor that into the dilution. And then we get the final dilution factor, which includes everything. But you can wait to the end and simply divide by the volume you put on the plate. So either way, it's the exact same result. So just showing you the procedure again. Your stock phage solution, 1 ml is being transferred to each tube. You thoroughly mix the tube and transfer 1 ml to the next tube, right? Mix, next tube mix, next tube mix. So, but all just 1 ml samples. All of this is in the water bath, along with your soft agar deeps. You then transfer 1 milliliter of each phage solution, starting with the original stock, into the corresponding soft agar deep. So you got your phage in the soft agar. Then you have to add 0.2 milliliters of E. coli B, into each of the soft agar deeps. Because again, the purpose of the soft agar is to mix the phage and E. coli together. Then you can pour that mixture on top of your TSA plate, gently swirl it, right, to spread everything out, and incubate your plates. And in next class, we will see results like this. So now I'm going to switch to the docx form so I can write in it or type in it. And I put both the PDF and the docx in Blackboard. So, but the docx you can type in, or, you know, with my writing pen, I can write more easily. So here we have our 5 plates, and these are labeled A, D, E, or A, B, C, D, E, and they go in this order, A, B, C, D, E, right? So a little strange to go this way. And so they've given us all the dilutions on the plates, right? So plate A, is 10 to the negative 4, right? And plate B is 10 to the negative 5. If you want, you can write 1 times 10 to the whatever, but since we understand it's there, we don't necessarily have to write it because we know it's there. Right, so what are the dilution factors?
10 to positive 4.
Yeah. Right, so just make the exponent positive, right? Okay, so that's easy. Now, number of plaques. So when we look at the pictures here, like our first plate, and actually the second plate too, what we actually see is mostly plaque, mostly phage. Very little E. coli on the first plate, and just a little bit scattered here and there. Otherwise, it's basically almost all plaque. So what would you call that?
TNTC.
Right, TNTC. For plate C, so guess what you have to do? You have to count.
Count each one.
Yeah, so when you have a lot like this, what I suggest doing is like, count half of the plate. So draw an imaginary line, for you guys. In real life, you could actually take a Sharpie and draw on the bottom in half and start counting, because if you reach 150, you know it's going to be too many, right? So, or you can count with me, right? So wherever you see a plaque, you make a mark. And actually we have colony counters, which we use to do this. And each time you tapped, it'd leave a mark on the plate like this and make a beeping sound and start counting. So before I get too far, see, what do we have so far? Right, we have 20. Looks like maybe one here.
20, 21, 22.
It's kind of hard with some of these.
I
That's
think
about
there'll be
100
more
right there.
than, yeah, I think
Huh?
there'll be more than 300.
Yeah, since that's about 100 there, this plate is also going to be right TNTC. So now we have plate D here, right? So we do the same thing. Oops. Ah. Yeah, some of these it's hard to tell if they're actually plaques or not. So let's just stop there. So how many did I get?
53.
Ah. And what about the last plate?
Probably too few to count.
Too few to count.
That's too few, right? So too few to count. But if I ask you, how many plaques would you expect to get on plate E? What would you tell me? How many plaques would you expect to be there, knowing that plate D has 53 plaques? How many would you predict would be on plate E?
Maybe five. 5.
5.
Right, between 5 or 6, right? So each plate, right, the difference between each plate is a factor of 10. So as we're becoming more dilute, each plate should have 10 times fewer plaques on them, right? So if there's 53 plaques here, we predict there should be between 5 and 6 plaques here, right? And sometimes this is exactly what happens. You know, our students, their technique is so good that even though the plate is too few to count, it actually has the correct number you would expect to be on here, right, based on the plate that's countable. So it is possible to get accurate results, right, doing this procedure. And sometimes our students actually do such a good job, you know, pipetting that they do get exactly what you would expect. So we have 53 plaques on plate D. Right, so remember it's number of plaques times the dilution factor. And what's the dilution factor for plate D?
10 to
10 to the positive 7.
Right, 10 to the positive 7. But of course, we know this is not proper notation, right? So how would we write it in proper notation?
5.3 multiplied by 10 to the positive of 8.
Yep. And of course, we always include units, right? So any questions on how we did this? Now we have questions. So why did you serially dilute the phage solution? So why did we perform this serial dilution in the first place?
to see how many phages infect the E. coli.
So to see what again?
How many bacteriophages infect E. coli?
Okay, so we needed to dilute, right, to get countable plaques, right, between 30 and 300, right? So just like when we diluted our bacteria, we needed to get countable colonies. So here we need countable plaques. So we have to dilute our phage so that we can get that. How do T-even bacteriophages like T2 or T4 differ from others? Well, you tell me, how are bacteriophages different from the other viruses we've talked about?
They have like a really complex structure.
Right, the bacteriophages are complex viruses, right? Most other viruses are not complex. What else? Some animal viruses have something these don't have. What's that? No lipid what?
So,
No lipid what?
Membrane.
Membrane, right? No lipid membranes. Right? Our even bacteriophages have DNA only, whereas other viruses may use RNA as their genome. What results would you expect to see if you had used a lysogenic phage? Well, we already answered that, right? What would you expect to see?
No change. It would just look like E. coli.
Well, just bacterial growth, right? And the bacteria would have grown, but that's all we would see is just bacterial growth, no plaques. Why is it important to obtain a confluent bacterial lawn in this exercise? So why did we need solid growth all over the whole plate? To see what?
To see the plaques.
We have to see the plaques, right? Because we don't want bare agar surface, right? Because we're looking for plaques. So in order to see them, we have to have bacteria growing all across the entire surface. Why did we use E. coli B specifically?
Because it's susceptible to infection.
Yep. It's susceptible, right, to infection with the T. even bacteriophage. Why did we use the soft agar?
Mix.
Right, to mix the phage and E. coli together, right? So bacteriophages by themselves are not going to grow on
Professor,
agar.
do you mean to mix phage?
And
You
E.
said to
coli
mix.
together. Or mix phage and E. coli together. So why do we need to use TSA plates or agar plates for this experiment? All right, we're trying to grow the virus, but why do we use the agar plates?
So the bacteria can grow. So the
Yep.
virus can get into the bacteria.
Yep, so, to grow our host cells, right, to grow the E. coli, right? They need to grow on agar, and we need the E. coli to grow the virus. So the answer that everyone now knows, will the plaques get bigger in size if we incubate the plates longer? Will the plaques get bigger? This one you all know. Will the phage keep multiplying and wipe out all the E. coli?
Yes
Really?
or no?
No, they will not, right? And so why again will the plaques not continue to grow and spread? What kind of cells do they want?
Oh, because the bacteria, they want them in log phase.
Right, they want log phase bacteria. I'm not going to type all that, but we already know that the plaques stay small because they want young, healthy, metabolically active cells in log phase. That doesn't last very long under ideal conditions, right? Why do we not use the TNTC or the TFTC plates? I mean, the TFTC, we can count the colonies, right? That's easy. Why do we not use that? Or use a plate that's over 300 plaques? It's not what?
Is it because it's not accurate?
Yep, it's not accurate, right? Above 300, the plaques all start to grow and merge together, so it's hard to see them as distinct plaques. And below, 30, it's generally not considered reliable because of experimental error. And to finish off with a problem. So read through the problem, and then I'll talk you through it. It'll take a couple of minutes to read through. So this is a little bit more complicated, right? We have multiple dilutions, but they're a little bit different, right? Not the exact same each time. So here we're starting with the phage, and we're going to be adding 0.1 milliliters into 9.9 milliliters. So what kind of dilution is that? If we add 0.1 over 9.9 plus 0.1, our total is 10, right? What's 0.1 divided by 10?
Ten to the negative 2.
Right, or 0.01, which is equal, right, to 10 to the negative 2. Right, so that's our first dilution is 10 to the negative 2. We repeat and do the same dilution again. We add 0.1 into 9.9, so it's another 10 to the negative 2 dilution, but we're already diluted 10 to the negative 2, and we do it again. So what's the total dilution here?
That'll be -4.
Yep. And then we do it again. Another 10 to the -2 dilution, but we're diluting a solution that's already 10 to the -4. And so this one becomes
-6.
-6. Now things change. Now we're going to transfer 1 milliliter into 9, right? So we're back to our standard 1 to 10 dilution. And we know that's equal to what?
To the 0.1.
Well, right, 0.1 or 10 to the -1, right? So -6 and -1 makes
-7.
-7.
yep, -7. Now the amount we're putting on the plate is 0.1 milliliters. So remember, there are two ways you can do this. When you get near your final answer, you can divide by 0.1, or we can include the 0.1 now. And so remember, 0.1 is equal to 10 to the -1, right? And -1 and -7 is what?
-8.
-8. So remember, that's the effective dilution on the surface of the plate is 10 to the -8. But the number we want is the dilution factor, right? And what is that?
+8.
And so we know there's 180 plaques on that plate, right? 180 times 10 to the 8. We have to put this in proper notation. And so what would it be?
$1.80 to the 10th.
Yep.
10 to the 10th. PSU.
Right? So it's just a problem involving different dilutions, right? Either 0.1 into 9.9, which is a 10 to the -2 dilution, or 1 into 9, which is a 10 to the -1 dilution. And so we calculated, right, the dilution in each tube. When we got to the plate, we included the volume we put on the plate in our final dilution here. Or you could have waited to the end and then divided by 0.1, and you would still end up with the same number. I just think it's easier to include it in our dilution on the surface of the plate. And then we just make that a positive exponent, multiply number of plaques times the x of the dilution factor, and then put it in proper notation. So questions on how we did this? So 1.8 times 10 to the 10, how big is that? What would that number be?
Is that like more than a billion?
Yep, more than a billion, right? So that's a lot of viruses per milliliter, right? Over a billion. And do you know what that broth would look like? It would be crystal clear. It would look like sterile broth, even though it's got billions of viruses in it. It would look crystal clear because the viruses are just too small. If you had a broth culture with a billion bacteria in it, you would see that because the broth would be turbid, right? It would be cloudy. You could easily tell there was bacterial growth there. But viruses are just too small. So even if there's billions of viruses per milliliter, the broth is crystal clear. It looks like it's sterile broth, even though it's loaded with viruses. Any questions? right, well, I don't know why your class is faster than the morning class. Maybe you guys need to ask more questions. Well, that's all we're doing today. Like I said, it took longer in the morning class. So any questions on gradient plates or the plaque assay? And no, I haven't seen lab exam 2 yet, so I don't know if there'll be a problem on there or not, but you should expect one, but I haven't seen it yet, so I don't know. But that's a long way away, right?
Yeah.
Did you say you've not seen it yet, Professor?
Yes?
Did you say you've not seen the lab 2 yet?
Nope. It has to be given to us. That's not going to happen for a while. So, but you should be prepared for at least a little problem on it. Nothing as complicated as this probably, but a little problem on it. Now, I had a student this morning, and so what if you gave me an answer like would that answer make any sense?
No,
No. And why not?
because they're not the same.
Well, right, but what is that number? 1.8 times 10 to the negative 10. What would that be?
It's teeny.
Huh?
It's like a tiny number. It's like
Yes,
0.0018.
it's 0.00000,
Below.
etc. Right? So it's a teeny tiny fraction of 1. And that kind of answer makes no sense, right? Because we're calculating the numbers of viruses. So it has to be a whole number, right? It can't be less than 1. And so, throughout your education, in your math classes, chemistry, biology, physics, whatever, have any of your instructors ever said, when you get an answer, look at it and ask yourself, does it look reasonable? Has anyone ever told you that? Anywhere, high school, college, wherever. Whenever you're calculating something, students just plug and chug, right? They crunch the numbers, whatever the calculator spits out, that has to be the correct answer, right? No. But you should always look at your final answer, and does it make sense? Does it seem reasonable given all the other numbers you've used, all the parameters, for the problem? Does your answer look reasonable? And that's something hopefully someone somewhere along the way has told you. Look at your final answer, and does it make sense, right? Does it look reasonable? Because if you wrote something like this, 1.8 times 10 to the negative 10, that's not reasonable, right? That doesn't make any sense. And so whenever you're doing calculations, you should always look at your final answer and say, does that make sense? And so I have another example, which some of you may remember from Biology 150. I know how much you love that class, right? You remember the experiment, the amylase experiment, where you did the maltose standard curve, where you measured the absorbance of different concentrations of maltose to make a standard curve. And then later, when you did your experiments and you got absorbance values, you had to use the standard curve to find the concentrations of maltose, right, that was created, right, from the amylase reaction. And so when you make the standard curve, the values of maltose were like, 0.1, 0.2, 0.3 milligrams per milliliter up to about 1, and that's it, right? So all the absorbance values are based on concentrations of maltose less than 1 milligram per milliliter. And so, when students got their, the equation of the line, and then they got their different absorbance values, and they had to plug them in to find the concentration of maltose milligrams per milliliter, I had several students who gave me values like 4.3 or 2.8, even though the standard curve only went up to 1. So how can you get a concentration above 1? And so that's an example of where you look at the answer and you say, does that make any sense? And obviously no, because you can't calculate something that's greater than 1 if your standard is based on values that are all less than 1, right? So it's just an example of how you should look at your final answer and ask yourself, does that make sense? Right? Because you don't just plug in the numbers and whatever the calculator spits out, that has to be correct, because often it's not. Right? So always check your final answer. Does it look reasonable? Right? How can you get a negative value if your line is going up? Right? So just in general, it's always good to look at your final answer. Ask yourself, does this look reasonable? Does this make sense? And that could help you in the future, right?
Yeah, it could, for sure.
Right. I mean, has anyone ever told you to look at your final answer and see if it's reasonable? Anyone?
Yeah, I think math teachers tell us that.
Yeah, math people should do that a lot. But hopefully in your science classes, you've also had that where someone has told you to look at your final answer to see if it's reasonable, because that's, it can be very beneficial, right? Because you get an answer that really doesn't make any sense, but hopefully you know enough to look at that and say, yeah, that can't be right. So anything else? Well, that's all I've got.
Thank you.
Thank you, Professor.
Okay.
Bye.
Enjoy your weekend and study hard.
Thank you. We'll show up on Blackboard at 12 on Monday.
Right. It'll start at 12, right, for you guys. Go until 1. Lab will start at 10 after 1 on Monday.
Okay, great. Thank you.
All righty.
Bye.
See you guys Monday.